Well, hello and welcome to chapter 17, all about protein synthesis. So, main idea is really right here. We have DNA, it transcribes, making mRNA, and then we go through translation, which basically makes a protein. Also illustrated here. So, what do genes code for? How does DNA code for cells and bodies? So really, your DNA is just sitting there. It's a code. It's those proteins that are made. So everything, all the instructions are in DNA. Those DNA make proteins, which really is what traits and effects that you see all throughout. So you'll often hear in biology the central dogma. Alright, so the central dogma, what that's talking about is that you always have to have DNA to protein. DNA can go through replication. It can be made, RNA can be made from it. That's called transcription. And from that RNA, we make proteins. That's called translation. Those, in turn, will make traits, what you see in yourself. But exactly, proteins are doing all the work. So, we get metabolic diseases, and it suggests that genes are coded for enzymes. So, we talked about a lot of disorders, and really what's going wrong is an enzyme. Each disease, the phenotype, what you're seeing, is caused by some gene product that didn't go right. Usually a lack of an enzyme. So here's your pathway. You have product A, enzyme, and keeps going on. That enzyme doesn't work, you get a disease. That enzyme doesn't work, you can get a different disease. This enzyme doesn't work, that's what, a different disease. And then you can get this one that work and get a different disease. So Tay-Sachs is one such these where the lipids are building up in the body. PKU is when your body can't digest phenylalanine, and so you have to eat a protein diet. Albinism, so the lack of melanin. So yes, you are just a sum of your proteins. A couple people that figured this out, Beadle and Tatum, 1941, 1958, they end up getting a prize finding out that one gene goes to one enzyme. How they did this is they actually took some fungus spores, grew them all up, and then found they tested so that here's a control. Right up here they created a mutation. They have positive control, growing it on the complete negative. Here's a negative control. And all the different things they experimented with. Different supplements, and you can see over here, the arginine, they found the mutation on there. So, what ends up happening is you have your blueprint all locked in safe, the nucleus. mRNA comes and makes copies of that blueprint, so that's called transcription. mRNA goes out into the cytoplasm, onto the ribosomes, and we make a whole bunch of amino acids, which in turn make proteins. And that's called translation. That protein will turn into the trait. So, transcription goes from DNA language to RNA. So some things about RNA, some differences between RNA and DNA is that RNA is made out of ribose sugar instead of deoxyribose. The nitrogen bases, it has uracil instead of thiamine. So you use U is going to pair with A, and C and G will still pair together. It's single-stranded, so here's your RNA, there's your DNA. And there's lots of different types of RNA: mRNA, tRNA, rRNA, siRNA. We have 3 kinds that help with protein synthesis. So ribosomal, really, what it does is forms ribosomes. And so it's made in the nucleolus. It has 2 small units, and again, this is the ribosome, small and large. And the interesting part is the bacterial ribosomes are actually a different size. And so you can see differences within them. So this is showing translation, tRNA is coming through, and the ribosomes going on the mRNA. And then you have your transfer RNA, and you can see it's made of a whole bunch of codes. You get this anticodon, and that is going to be who pairs up with the mRNA. There's also an attachment for amino acids. So transfer RNA have an anticodon, and there's also something called the amino acyl tRNA synthetase, which will attach the specific amino acid using energy from ATP. And we have messenger RNA. So messenger RNA is built from DNA and carries that to the ribosome. So during transcription, we have the template strand, and so here's our template strand, and then we have this one that's just sitting up there called the coding strand. And so that's going to have the same sequence because, makes sense, CGTA is going to pair with GC, so since that also pairs with the coding strand, they're going to have the same sequence. Synthesis or making of RNA happens in the transcription bubble. The enzyme that does this, big pink blob here, is my RNA polymerase. And we build it from 5' to 3'. There's 3 different RNA polymerases. So you have polymerase 1 that only transcribes R RNA genes, so they make the ribosomes. Polymerase 2, this transcribes genes to mRNA, so this is a biggie. And we have polymerase 3 that transcribes tRNA genes. Each one has a specific promoter sequence, which we'll talk about in the next slide, but that's who starts the gene that they recognize. So how do we know it's red? We have a promoter region. So nucleotides. So this is the binding site before the beginning of the gene. One such of these is called the TATA box. So you can see it's named for, it's always at TATA. And that's the binding site for RNA polymerase and different transcription factors. You also have an enhancer region, which is upstream, and it's the binding, it turns the transcription on high. You have different transcription factors, one such as the initiation complex. Those transcription factors are going to bind to the promoter region. It's kind of white blobbish area. And these transcription factors are proteins that help it bind to DNA. They could be hormones. And they help turn on and off transcription. This whole initiation complex is going to trigger the binding of RNA polymerase to DNA. Next up, we have to match the RNA bases to DNA. So RNA will help doing that, but we have our A to T, A to T to A to U, G to C, and so on. 5' to 3', and it goes on. But one thing we have found is that we have a lot of junk. So we can't just copy everything. Eukaryotic genes are not continuous. The exons are the real gene. So the exons stay in, and they're called exons because they're expressed. So that's the coding DNA. So that's the blue. Then we have introns, which are the junk, and they're in between sequence. And you'll see that those will be taken out. So introns come out, exons stay in. So mRNA has to have some editing before, so it gets copied from DNA, but it needs some editing. So introns are removed, and so all these all get removed. Exons are expressed. How that happens is called mRNA splicing, kind of like slicing. So you have post-transcriptional processing. So it's gone through transcription, it hasn't left the nucleus yet. It needs some work, and before it goes through this editing, it's called pre-mRNA. We edit out the introns, get rid of them, and we get the mature mRNA transcript. I can't really see them up here. So there's eukaryotic DNA, the transcript, which is called pre-RNA. You can't see it. Let me see. And here's your spliced post-transcript mRNA. These are the people who figured this out. That's smart people. So splicing has to be accurate. I can't oops, got a little too many nucleotides. One single base can throw off the entire reading frame. So if we have all of these, so they're read in threes called codons. And if I lose one, and so I got rid of the red part, there goes the intron. But if I accidentally add, so that's same piece, I just divided in three for my codon, I have we'll end up getting these amino acids. If I do the same thing, but I accidentally keep a G in, now my whole reading frame is gone. You can see here, instead of the CGG here, okay, it's still CGG, but now instead of UCC, I have GUC. And oops, that's completely messed up. So we have SNRPs. They're called small nuclear RNA, type of protein. And they're going to help us with this. So basically, you get this spliceosome, which consists of SNRPs, and they'll recognize that splice sequence, and they help cut and paste the gene. So basically, you get all these SNRPs. We're going to come around, recognize the sequence, and get rid of the sequence. Now we can have our exons together. So we have with the processing, there are sometimes using ribozymes, which aren't soy proteins, they're actually RNA molecules. And so some pre-RNA can remove some of their own introns. There is also alternative splicing. I don't know if I'm going to get it. And so they're produced from the same gene, but sometimes the introns aren't always introns, and they'll leave them in. And depending on which gene it is. So here, left it in, this one left a different gene in. And then we have to protect it, otherwise there's enzymes that will attack it, and so you have to add the G